Hello students, how are you all? You know what? I uploaded a video on carbohydrate metabolism. Actually, that was a two-part video, and somehow due to some technical difficulty, I failed to upload the part one. It was a two-part class which I took live online, okay, and uploaded the second part. So there were a lot of comments of students who watched the second part and who actually asked me to upload the part one. Okay, so it's okay. So here it is. So today we'll be discussing what we actually discussed in the part one of the live class. Okay, so what was the class about? Actually, the class was about essentials of carbohydrate metabolism. Okay, now to understand the essentials of carbohydrate metabolism, first you need to understand one thing, that is the competencies. Okay, so first you need to look at the competency. So competency 3.1, okay, as well as competency 3.2. the total competency number 3.13 actually deals with the metabolism of carbohydrate, both chemistry and metabolism. The chemistry part is mainly in the 3.1 and rest are in 3.2. So you need to look at the competency in order to get an idea about your specific learning objectives. So what you need to know, you can see over here. You can first, if you have not downloaded this, download it immediately under UG curriculum volume 1 NMC, National Medical Commission. If you just type it in the Google, you will be able to download this. Okay, so over here you can see there are multiple things that are mentioned. Okay, so often students get lost, okay, if they don't attend classes attentively. You know what I mean. Okay, there are good students and there are not so attentive students. Everyone is good. Okay, since you have cracked NEET, I believe you are good. But it happens, especially during online classes, we tend to lose focus, and then when studying from the textbook, we cannot often relate what actually we need to learn and what is there in the textbook. Okay, so today what we'll be doing, what I actually did in that class is I simply took a textbook, reference textbook of Satyanarayana, okay, and I asked my students to simply mark those areas. Okay, this is basically a self-directed learning SDL course where I am actually your mentor and I am helping you to go through the textbook, and I will exactly tell you what part you need to study and what part you need to omit, because there's a lot of time. I mean, there's absolutely no time for in which you need to cover all these three subjects: anatomy, physiology and biochemistry. And for anatomy you need to allot 60% of your time, right? If you don't, then it will be very difficult, because even being a teacher in biochemistry, I always tell my student to first focus on anatomy, and then in the remaining time you should divide it into physiology and biochemistry. Okay, enough about the lecture. I mean, enough about the advice. Okay, the lecture will still continue for another, let's see how what is the time I'm able to wrap it up. And you know, after I end this video, you can watch the previous video which is already uploaded a few days back, that is essentials of carbohydrate metabolism part 2. Okay, so this is part 1, and my reference textbook is U Satyanarayana, the textbook of U Satyanarayana. And believe me, if you are studying something else, I mean, if you are studying Vasudevan, if you are studying Rafi MD or AC Deb, whatever is the SK Gupta, any textbook, the outline of this class will be the same. And please note down the topic, and while following along, I will also tell you what part you need to focus and what part you need to omit. Okay, see, so here are few pathways in the textbook of Satyanarayana that is highlighted. Okay, so you can see it's written glycolysis, Krebs cycle or Tca cycle, gluconeogenesis, Glycogenolysis, Glycogenesis and HMP shunt. Okay, so what's the deal about all of them? Okay, my slide is glitching, right? So the deal is, and in part 2 I already covered uronic acid pathway, galactose, fructose, etc., right? And I also covered a part of glycogen metabolism in part 2. The common thing that you need to study from all of them, okay, let's first tell you what you don't need to study. There is no room to study each and every step one after another, remembering every enzyme, every cofactor. Yes, if you are interested, you can study. Believe me, of course you can. If you give time, you can study. You can remember for 1 day, 2 day, but trust me, after you are burdened with everything, after the full syllabus comes to haunt you, you will forget everything. So from the day one, you should read smart, okay? And in Viva, we examiners, we tend to ask those smarter areas only. So even if you give your whole lot of time to study everything, unless you revise everything and you tend to remember everything, it's a waste of time. So please study smart only. So in order to study these topics smartly, what you need to know, okay, there are few acronyms that I will use, okay. Number one, I will use all, or you can also simply write this in your textbook or write in your notepad. So I will, whatever I will say, you just mark it in your book with pencil or a marker pen, whatever you want, okay. When I say all, okay, what do I mean by all? Suppose in glycolysis, I will say all. It doesn't mean you need to read from glycolysis, say page number 238 to 242, everything, page after page. No, it's a waste of time. So what you need to read? Number one, you need to read the rate limiting enzymes or the rate limiting steps or irreversible enzymes. Excuse me. So these are the important things that you need to know in case of glycolysis, okay. Rate limiting steps or irreversible steps. If you look at the process of glycolysis, there are 3 rate limiting steps, okay. I'm not going into much detail, but of course, in order to, for example of glycolysis, I need to tell you, number 1 step is hexokinase, number 2 step is phosphofructokinase 1, and the next irreversible step is pyruvate kinase, PK, okay. So regarding these 3 steps, you need to remember what are the reactants and what are the product. I mean, from what it is converted to what, okay. And also you need to know how to reverse these steps. When you learn how to reverse these steps, basically you are already learning about gluconeogenesis. This is how the cycle works, okay. We'll be showing you, definitely will be showing you, but just for glycolysis, what you need to remember, you need to remember the rate limiting steps. What are the enzymes that are involved in the rate limiting steps? What are the reactants and products? How you can reverse those rate limiting steps? I mean, what are the enzymes that can reverse the reaction? Otherwise, all other reactions are bidirectional. For example, the aldolase reaction. Aldolase can convert product A to B, and the same aldolase can convert the product B to A. For example, phosphohexoisomerase, it can convert glucose 6-phosphate to fructose 6-phosphate and vice versa. But hexokinase, PFK1 and PK, they don't, okay. So number 1 is the enzymes, all about enzymes. Number 1 and 2 are about enzymes, right? Number 3 is about energetics. Energetics of glycolysis means we need to learn about the steps where ATP is consumed or it is produced, and also the steps were reducing equivalent, okay, because NADH is also ultimately equated in terms of ATP, okay. So the steps in which ATP is produced or utilized, and the steps in which NADH or FADH2 are produced and utilized. So either ATP or any reducing equivalent is produced and utilized, we need to know that, okay. And lastly, the regulation of glycolysis, okay. So in case, if I say all, in general, this is the thing you need to learn, okay, before everything else. Step after step, you don't need to know, only the irreversible steps or the rate limiting steps, how you can reverse those steps, especially in case of glycolysis. In other pathways, it is not needed, okay. This is special in case of glycolysis and glycogen metabolism. Other pathways, you don't need to know the reversing enzymes. So rate limiting enzymes or irreversible steps, energetics, that is how many ATP or NADH are produced and utilized, and the regulation of the step, okay. In regulation also, you need to give under few headings which we'll be discussing very soon, right? So just see, okay, I'll keep it. Just before the start, I am overwhelming you with so much information, but as you follow along, you will be see the pattern is actually really very easy to understand, right? So let us look at the book. So in case of, so first, for if you are using this edition, this is more or less the same. Satyanarayana, the textbook, doesn't undergo much changes. Okay, so to start with, entry of glucose into the cell, that is absorption of glucose, you need to know short note. Okay, remember, short note about glucose receptors. Okay, glucose receptors are of two types. So just add beside this page, okay, over here write SN. Okay, short note about glucose receptor. So glucose receptors are of two types. Number one is sodium dependent glucose transporters. Okay, sodium dependent glucose transporter, in some books it is written as SGLT, and in some book it is written as S-GLUT. Okay, sodium dependent glucose transporter, right? Apart from that, there are few receptors which are not sodium dependent, but they are simply glucose transporters. They are also named as GLUT. So what you need to know, what are the location and function. That is only one function of each receptor. Okay, so sodium dependent glucose transporter, where are they located, and the nature. Okay, nature of this transport, whether it is an active transport or passive transport or facilitated diffusion, that you need to know, because this will also come under come handy when you are answering the chapter of bioenergetics. Okay, in that chapter your teacher will be asking you all these questions. You know what, in Viva a teacher don't specifically ask you chapter by chapter. Okay, he asks you back and forth. So from bioenergetics he can enter into carbohydrate metabolism via GLUT receptor, and from carbohydrate metabolism he can actually go into bioenergetics via this glucose receptor. So these are very important. Often students tend to learn glycolysis, Krebs cycle, everything, but examiner actually can give him some trouble but just by testing his knowledge regarding the GLUT receptor. So for example, GLUT1 where it is present, GLUT2 where it is present, GLUT4 where it is present, you just make a chart in your own note, and just in the short note you can give that chart. You will get full marks. You don't need to write pages after pages, because examiners always prefer objective type of answers. Okay, mark my words, examiner always prefers objective, right? So in your textbook, if a chart is already given, it's fine, or else you need to, that's a part of your homework, you need to make an own chart where GLUT1 is present, where GLUT4 is present, like that. Okay, for example, GLUT1 is present in erythrocyte, that is RBCs, GLUT4 is present in skeletal muscle and fats, that is adipose tissue, so on and so forth. So just please look into it. Okay, so now the first major pathway that we need to study, glycolysis. Again, in glycolysis you give a double tick mark and you write all. Okay, and by all, again let me tell you, all doesn't mean you need to read pages after pages. You need to write, you need to study about these things only: rate limiting enzyme, energetics, and regulation. Okay, fine. So glycolysis all, and also there are some things that I will tell you in between, that where glycolysis takes place. Glycolysis takes place in cytosol. Okay, this of course you already know from your +2 knowledge. Okay, I don't need to tell you glycolysis occurs where. Okay, the regulation part I'll be definitely telling you, and I have already made video on regulation in carbohydrate metabolism. Okay, if you cannot find that video, please let me know in the comment section. I will again I give you link to that video. Basically it's the function of insulin and glucagon and how they play a reciprocating role in regulating carbohydrate metabolism. Very easy to understand and very interesting. Once you understand, all regulation will be very easy to answer. All regulation type of question, not only in carbohydrate metabolism but in all metabolism. Okay, anyway, okay, I think after the class is over I can search my channel and I will show you where the video is located. Okay, for now let us move on. So you don't need to read pages after pages. Okay, splitting phase, investment phase, fine, you can just know the name, energy regeneration phase. Often in Viva, when you are just giving Viva in carbohydrate metabolism chapter, that is the part completion, examiner may ask you all these things, but just by knowing the irreversible steps you are actually done. Okay, examiner will not penalize you if you cannot name energy investment phase or splitting phase or energy regeneration phase. What we'll be more interested in is the regulation of carbohydrate metabolism. Okay. Anyway, so one thing you need to note, that is one inhibitor of glycolysis. This is very important. So underline these two and you write viva. Okay. So iodoacetate and arsenate, you just underline these two and you write viva. Beside this, because this will be asked in viva as a short question, okay, during oral examination. So this part is very often students tend to miss, but if you can answer, examiner will be very impressed. You can see over here, this is the chart that is given in every textbook. All the arrows are having two sides, but the arrow of hexokinase or glucokinase is a big bold arrow in a single direction. Same is with phosphofructokinase, this is one, and same with pyruvate kinase. Okay, so the 3 irreversible steps I mentioned that you need to know, and also the steps where the ATPs are produced and generated, okay, that you also need to keep in mind. Okay, so total ATP calculation from one molecule of glucose, if it undergoes glycolysis, both in aerobic and in anaerobic scenario, how much ATP it will generate. Okay, so there you go. Next, in case of, just remember, in case of anaerobic glycolysis, that is why if the oxygen concentration is low, pyruvate will be converted to lactate by the enzyme LDH or lactate dehydrogenase, and in that case energy will be utilized. See, NADH is converted to NAD. Okay, this happens in active working muscles and in hypoxic condition. Fine. So that's an explain why, and I have already mentioned it multiple times. You can find there are many explain why type of question. I have made videos about many explain why type of question. Over there is a question, why glucose estimation, why the vials used for glucose estimation contain sodium fluoride. Okay, there I have already explained in detail why it happens, but you can see the answer is given here. Okay, what is the answer? Basically, see, glucose estimation means we need to estimate the amount of glucose that is present in this cc of blood. If I give my blood in order to test whether I am diabetic or not, we are trying to measure the glucose, right? So what happens in that blood? Okay, there is RBCs, and RBCs cannot utilize glucose in any other pathway except glycolysis, because they don't have mitochondria, and the process which occurs in cytosol is glycolysis. So if we can somehow stop glycolysis, then the glucose will not be utilized and glucose will remain intact. What is the problem if glucose is utilized? They will give false low value. Suppose my blood glucose is 100, okay, or it is 120. Then if it remains for 2 hours or 3 hours just in the vial, the glucose level will fall if we cannot stop glycolysis. So it will give wrong value. So in order to prevent glycolysis, we give an inhibitor of glycolysis. Mind it, we also discussed there are multiple other inhibitors of glycolysis as well. But why we are choosing sodium fluoride? Number one, because it is cheap. It does not inhibit with the glucose assay. Okay, and it also sometimes serves as a dual function of anticoagulant. Okay, anyways, just know this, sodium fluoride is used in order to prevent glycolysis, in order to inhibit glycolysis, and that will prevent the glucose from coming down by utilization in the RBCs. Okay, so there is one explain why type of question. You can write explain why or justify. You just mark this paragraph and write, give a second bracket and write justify beside this paragraph. Okay, fine. So then we move on. You can see production of ATP in glycolysis. This is very important. I already told you, right? This is the calculation of energy in glycolysis, and this is the chart where it is given very nicely. Okay, of course we also need to know citric acid cycle as well, but we are coming to citric acid cycle in a few moments. But for glucose, you see, this is the total energy calculation. Believe me, this will be asked in viva, and you need to mention what are the steps in which ATP is utilized or it is produced, and what is the net result in ATP calculation. Okay, you see in your textbook there might be given two numbers, one is 6 and one is 5. Why is that? Because there are two existing theory. Basically, this is why this is there. NADH produces ATP, energy that is equivalent to ATP, right? And how it does? It does by undergoing or by participating in few shuttle mechanism, that is glycerophosphate shuttle or malate shuttle. Okay, now the old theory was there, that is one NADH produces 3 ATP, that is equivalent energy of 3 ATP. Whereas the new metabolomic, new energetics research papers have suggested that one NADH produces 2.5 ATP. So over here you can see 2 NADH is producing either 6 or 5 depending on the old and new calculation. Similarly, like FADH2, old theory was FADH2 was producing 2 ATP and new theory says FADH2 produces 1.5 ATP. So it depends on what you take, but you have to mention the basis of my energy calculation is this. That is what is written, brackets in red color represent ATP synthase as per the new P:2O, that is phosphorus to oxygen ratio of ATP, that is 2.5 for NADH and 1.5 for FADH2. For undergraduate students, both are right provided you know. Okay, but you cannot mix it up. In one step you are saying 3 and in the other step you are saying 2.5. If you mix it up, you are wrong definitely. So just stick to one and mention to the teacher, sir, the teacher who taught me in the class, he or she mentioned this thing. Okay, so we move on our next. See, irreversible steps in glycolysis. Of course, this is very important. You need to know this. And this diagram is important because it tells you about the regulation of glycolysis. Okay, so regulation of glycolysis you need to know for oral viva. We move on. This is extremely important. Mind it, this will help you in order to fetch honors marks if needed. That is role of fructose 2,6-bisphosphate in glycolysis. I have made a dedicated video regarding that. This is a 5 to 10 minutes video. Just need to know how fructose 2,6-bisphosphate is produced, what is the role played by phosphofructokinase 2. Those are important. Okay, so basically all of them help to regulate the phosphofructokinase step, that is the PFK step. So these step, if you go into much detail, is actually regulated by all these things. Okay, you can read from the textbook or you can just consult my video over there. I have simplified it so that you can easily understand this. Okay, next, this one, Rappaport-Lubering cycle, is just for oral. Okay, often short note may come, but just give it a read in case of if you're answering very well, then examiner may ask both in physiology and biochemistry, what is RLS, that is Rappaport-Lubering shunt or Rappaport-Lubering cycle. You should be knowing that 2,3-BPG plays a good role in oxyhemoglobin dissociation, that is dissociation of oxygen from hemoglobin and shifting of the curve to the right. Over there, this Rappaport-Lubering shunt will come, and then a physiology examiner might tell you, okay, you have read in biochemistry, so where you have read, then you need to say, ma'am or sir, I have read in glycolysis. Okay, next, Pasteur effect and Crabtree effect. These are very easy, but I won't suggest you to read. I have made videos in which I have actually explained the Pasteur and Crabtree effect. If you are interested, then only go through, but if you are in lack of time, you can simply skip these. Okay, you will not be penalized if you cannot say Pasteur and Crabtree. Okay, yes. So the next part, the next big question that is coming in your way is the pyruvate conversion. So glycolysis leads to conversion of glucose to pyruvic acid or pyruvate. Okay, and the next important reaction is Tca cycle. So Tca cycle starts with acetyl coenzyme A. So there is a reaction which links the two, that is known as link reaction. Okay, link reaction is also known as pyruvate dehydrogenase complex reaction or PDH reaction. Okay, one question you need to know about link reaction, and that is all. That is, what are the enzymes and coenzymes of pyruvate dehydrogenase complex, or what are the enzymes and coenzymes of link reaction? Okay, that's it. You don't need to read anything else. Okay, so over here you can see it's all written. The three enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. These three names you need to know. Okay, and you need to know the name of 5 coenzymes. Okay, remember this, students goofs up in viva, and if you can tell this confidently, you get a majority of marks, right? So what are the cofactors? It's written clearly, TPP, that is thiamine pyrophosphate, okay, vitamin B1, okay, lipoamide, coenzyme A, FAD and NAD plus. Okay, these 5 cofactors and 3 enzymes, or 3 enzymes and 5 coenzymes you need to know. Then you are done with link reaction. You don't need to read anything else. Okay, remember, I am telling you don't need to read anything else in perspective of whether you want to pass, and you want to pass with a decent mark and you have got a very less amount of time to revise. Okay, of course, if you want to learn the subject, you can feel free to go through everything. You can feel free to ask me question. You can go through detailed videos in my channel and you are sorted. Okay, but often the scenario is not like this. In last minute we tend to cover most of the things and read smartly, right? So again, regulation of Pdh complex is actually very easy. If you just look at this diagram, it may feel very intimidating, but if you look at my video where I have explained the mechanism of insulin and glucagon, how it controls anabolism and catabolism, then it will be very easy. Yes, regulation of Pdh complex may come in Viva, may be asked in Viva, and if it comes in theory, you can just give this diagram and you are done. But in Viva, the first question that will be asked to you is what are the enzymes and coenzymes. If you have answered them, then you need to worry less, even if you can't tell about the regulation. Okay, you get me? So we can move along. Next, we go to citric acid cycle. So what we need to know in citric acid cycle? Okay, do we need to know step by step the whole cycle? See, to be honest, glycolysis and Krebs cycle are the ones that are actually taught since your +2 days. Okay, if not +2, it is taught from class 7, 8, 9. Okay, I think now in new curriculum, CBSE curriculum, it is even taught in class 5. Right? So yes, if you remember everything, that is fine. Okay, but if you are lacking in time, which I believe you already are, or you will be when the exam is knocking at the door, then you need to know again the irreversible step. See here, citrate synthesis is an irreversible step. Okay, next, alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase and succinate thiokinase. These 3 steps are irreversible steps. So this you need to know, that is, in these steps, what are the enzymes involved, what are the reactants and what are the products. Okay, then there is no question of knowing reversing enzymes. We are not trying to reverse citric acid cycle, right, in undergraduate at least. Next, you need to know in which step reducing equivalents are produced, that is ATP, right? So in citric acid cycle, please know that direct ATP is not produced. NADH, FADH to GTP, etc. these are produced. Okay, so you need to know those steps. So the step in which NADH is produced, okay, there are 2 steps in which NADH is produced. There is one step in which FADH2 is produced. You need to know this enzyme. Okay, see here also NADH is produced, and also you need to know the step where GTP is produced. Okay, NADH2, you can answer this all 3 dehydrogenase steps. NADH is produced. It is easy to remember, right? Isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase, right? Except succinate dehydrogenase, where FADH2 is produced. Okay, remember, if it is succinate dehydrogenase, there is something wrong about succinate. Okay, you can remember like this. In case of succinate dehydrogenase, FADH2 is produced, and in case of succinate thiokinase, GTP is produced. Okay, so you need to remember all this. So you just mark these steps and remember these steps only. If you remember them, then you are done in knowing the reactions of Tca cycle. Next, you need to know the energetics of Tca cycle. Of course, how many ATPs are produced if one molecule of glucose is metabolized via glycolysis and Tca cycle. Mind that the amount of ATP that is produced from one acetyl coenzyme A, you need to multiply it by 2, because acetyl coenzyme A, that is pyruvate, is a 3 carbon compound and glucose is a 6 carbon compound. So one glucose leads to production of 2 pyruvate and 2 acetyl coenzyme A. So whole Tca cycle calculation you need to multiply by 2 if you are calculating from one molecule of glucose. Okay, so one question that will be asked in vitamin chapter, okay, after you have covered the vitamin metabolism, that is, what are the vitamins that play major role in TCA cycle? Okay, that's nothing very easy. See, TPP is a cofactor of both pyruvate dehydrogenase complex and alpha ketoglutarate dehydrogenase. Mind it, alpha Kg dehydrogenase and PDH, that is pyruvate dehydrogenase, they act in absolutely similar way. Both have got the other two enzymes, that is dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase as their cofactors. Okay, and each of them has got those 5 coenzymes, that is TPP, lipoamide, coenzyme A, FAD and NAD. Okay, so when it comes, FAD is a cofactor of succinate dehydrogenase, also alpha ketoglutarate. So FAD is vitamin B2, that is riboflavin. Niacin again NAD. All dehydrogenases are NAD dependent, and pantothenic acid, that is coenzyme A, is also involved. Okay, so these 4 vitamins are involved. So just you tick this and write oral or viva. Okay, next you need to know one inhibitor of Krebs cycle. Just one inhibitor will do for your viva. You can either write fluoroacetate or arsenite or malonate. Okay, I will prefer to say malonate. Okay, malonate inhibits the enzyme succinate dehydrogenase. Okay, fine. So that's all about TCA cycle. Yes, you need to know the regulation of TCA cycle, but that will again become very easy. Okay, if you understand what are the how insulin and glucagon plays a role, right? Next, in oral, okay, this question will come in oral or as a part of short question, not a long question, but a short part of a long question, or as a short answer type question. That is, what are the anaplerotic reactions in TCA cycle? Anaplerosis means, see, if a cyclic reaction happens, if a product is getting converted to another product, okay, eventually it will happen that some of the intermediates may be utilized completely. Okay, of course you may ask, okay, since it is a cyclic reaction, one product is already generated, but it doesn't happen that way. There are other side reactions as well. So we need to replenish the products of TCA cycle, that is the intermediates of TCA cycle need to be replenished somehow from outside. Those reactions are anaplerotic reactions, and there is a figure 13.10. Okay, see, the reactions concerned to replenish or fill up the intermediates of TCA cycle are called anaplerotic reaction, anaplerosis. These reaction, or just the names, or what are the anaplerotic reaction, or what are the products that are replenished, you just need to know 2 examples of anaplerotic reaction. So not all. If you just remember 2 examples of anaplerosis, you are done. Okay, remember TCA cycle acts in both catabolic and anabolic ways. Okay, there are products that are synthesized from TCA cycle. Okay, if you go into other metabolism details, you will see the intermediate of TCA cycle actually leads to formation of some product, that is the anabolic part of it. And TCA cycle in fact actually is breaking down glucose to produce energy, so that is the catabolic part. That is why TCA cycle is known as amphibolic. Okay, so amphibolic nature of TCA cycle, you need to just mention what something is produced from TCA cycle, and something is TCA cycle is also breaking down glucose to produce energy, right? So this is all about TCA cycle. Okay, we don't need to go into much detail about TCA cycle, nothing more you need to read. Okay, next is gluconeogenesis. This gluconeogenesis is actually very easy if you have studied glycolysis properly. Okay, because gluconeogenesis is simply reversal of glycolysis. So how is that? Okay, let's look. If you look at the pathways of gluconeogenesis or neoglucogenesis, both are same. Okay, you will see that it is nothing but a reversed glycolysis. Okay, nothing but a reverse glycolysis. You see everything. This is just glycolysis which starts from the opposite side. Okay, in Satyanarayana it is actually given in a student friendly manner. Basically it is showing that as if gluconeogenesis starts from glucose and it ends in pyruvate. No, gluconeogenesis starts with other substances, that is pyruvate, and then ends up in glucose. We are producing glucose. Gluconeogenesis, we are producing glucose, we are producing new glucose from something new. Okay, So basically all the steps are same like glycolysis, but just the 3 steps that were irreversible, we need to know how those 3 steps are reversed. So in glycolysis, if you already study about how these 3 steps are reversed, you already know about gluconeogenesis. Or if you just happen to skip those parts in glycolysis, now you are learning in gluconeogenesis. Okay. So the hexokinase step is reversed by glucose 6-phosphatase. Okay. The phosphofructokinase step is reversed by fructose 1,6-bisphosphatase. Okay, and the pyruvate kinase step is actually a bit complicated to reverse, so we need 2 enzymes to reverse them. One is pyruvate carboxylase and one is phosphoenol pyruvate carboxykinase. So this enzyme is abbreviated as PEPCK, PEPCK. Okay, PEPCK, phosphoenol pyruvate carboxykinase. So we need glucose 6-phosphatase to reverse the first step. We need fructose 1,6-bisphosphatase to inhibit the PFK1 step, and we need 2 enzymes, that is pyruvate carboxylase and PEPCK, to reverse the pyruvate kinase step. So if you know these 3 enzymes, these are the irreversible enzymes of gluconeogenesis. You need to, and if you know already glycolysis, you can, when gluconeogenesis happens, we are concerned with production of glucose, not ATP. So definitely this is not an energy efficient process. So energetics of gluconeogenesis will not be asked to you. What will be asked, that is the regulation of gluconeogenesis. The regulation is exact opposite to that of glycolysis. For example, insulin is promoting glycolysis, insulin will inhibit gluconeogenesis. Gluconeogenesis is promoted by glucagon, but glucagon actually inhibits glycolysis. So it is simply reversed. So what you can study is, you need to prepare a long question that answers the question, that is how glycolysis and gluconeogenesis are reciprocally regulated. This is extremely important. Okay, reciprocal regulation of glycolysis and gluconeogenesis are extremely important. That is the thing that you need to know, nothing else. In case of gluconeogenesis, just know the reciprocal regulation and the steps by which glycolysis irreversible steps are getting reversed. You can simply omit all these steps. Okay, see, lactic acid cycle or Cori cycle is actually you just give a tick mark and write R. Okay, that is just read. If the question, if Cori cycle come in the question, it is presumed to be a difficult question, because students don't tend to read it often. So if you are getting time to read Cori cycle, it means you are already well prepared. What I mean to say is, you can omit Cori cycle if you don't have time. Okay. Basically, it is how lactate produced in muscle, okay, is shunted to the liver, and it acts as a precursor for gluconeogenesis. Okay, that is the thing. Okay, this is the Cori cycle. So lactate that is produced in the muscle, it is shunted or transported to liver, and over there it undergoes gluconeogenesis to form glucose, and then glucose again comes in muscle, and the cycle goes on, because in muscles glucose is converted to lactate by anaerobic glycolysis. So this is a cyclical pathway. It is actually very easy to understand. If you have got some time to read, just give this diagram. If you give this diagram in Cori cycle, you get 80% of the marks. You don't need to elaborate anything more. Okay, so regulation of gluconeogenesis. Okay, it's again just hormones, influence of glucagon and availability of other substrate. You can just give it a read. Okay, I think from here I already covered in my part 2 video, but okay, I'll cover it in brief. Glycogen metabolism. Again, glycogen metabolism, you need to know that is important enzyme, that is a rate limiting enzyme. There are 2 rate limiting enzyme in glycogen synthesis, that is glycogen synthase and branching enzyme. And when glycogen is broken down, you need to know 2 rate limiting enzyme, that is debranching enzyme and glycogen phosphorylase. Okay, so in case of glycogen metabolism, you need to know 2 things. Number 1, the enzymes in each step, that is the rate limiting enzymes in glycogen synthesis and the rate limiting enzyme in glycogen breakdown. Okay, and you need to know the reciprocal regulation of glycogenesis and glycogenolysis. Okay, that is how glycogenesis and glycogenolysis are regulated together. So when one is formed, the factors that are helping in glycogen formation of course inhibits the factors that are formed, acting in glycogen utilization. For example, insulin helps in glycogen synthesis, but insulin prevents glycogenolysis, whereas glucagon helps glycogenolysis and glucagon prevents glycogen synthesis. So basically, if you know the hormonal angle, you are done. One thing you need to know, what is glycogenin. Okay, this glycogenin is basically a protein molecule that acts as a primer or the starting point in glycogen synthesis. You know what is glycogen? Glycogen is carbohydrate residues that are added one after another. Okay, but you simply cannot add carbohydrate residues to one glycogen. Okay, it does not happen. We need a starting glycogen molecule, a small molecule of glycogen, to which these enzymes can add carbohydrate molecules to make the glycogen molecule longer. Often what happens, to start with, either we need a small glycogen molecule or this protein glycogenin, they can accept glucose. And how the glucose is? Can glucose simply go and join? No, glucose needs to be activated by combining with uridine diphosphate. Okay, see, uridine diphosphate or UDP. So first glucose needs to combine with UDP to form UDP glucose. Okay, and then this UDP glucose will again break down and UDP will be gone, and this glucose molecule that is shown in black will be added one after another to the existing glycogen primer or glycogenin in order to produce a bigger molecule of glycogen. Okay, remember the glycogen synthase enzyme adds glycogen in a straight manner, that is, these are known as alpha 1,4 bonds. Whereas whenever we need to add branch, glycogen is actually branch molecule, it's a tree-like structure, that is alpha 1,6 bond, we need branching enzyme. Similarly, when glycogen is breakdown, you will see the debranching enzyme will cut at the branch and the glycogen phosphorylase will cut the straight ones, that is alpha 1,4 bonds. Those are actually very easy once you have understood the concept. So you don't need to read anything in detail about glycogenolysis other than what I have told you, the enzymes and how both of them are regulated. Okay, so yes, this is the allosteric regulation of glycogenesis and glycogenolysis. This figure is very important, you need to know this. Okay, this is allosteric regulation, not hormonal, but this is important. What are the substrates that are controlling regulation of glycogenesis and glycogenolysis? If your figure in your book looks different, then you can just give that figure, but just learn one figure from one textbook so that you can draw that in the answer script. Okay, so reciprocal regulation of glycogenesis and glycogenolysis. See, this thing is actually a bit difficult. The diagram, the diagram Satyanarayana, where there are two different diagrams given in case of one glycogen synthesis and glycogen breakdown, both are difficult. You just make your own diagram if possible, in which how both the pathways are controlled with the help of one set of hormones, that is insulin and glucagon. You can look into my carbohydrate metabolism regulation videos and then it will be very easy to understand. Next, in case of oral, examiner might ask you what is futile cycle. Futile cycle means if the pathway, that is one synthase and degradation pathway, are coupled. For example, glycolysis and gluconeogenesis, or glycogenesis and glycogenolysis. If both of them are occurring together, then it becomes a futile cycle, useless, right? Because something is produced, again it is broken down. So futile cycles are those cycles basically if they operate at same extent, then there will be no work. So it's hormonally controlled in such a way so that only one part is active, either the catabolism part or the anabolism part. If we combine them both, then it becomes useless and it is known as futile cycle. Okay. Next, we need to look at glycogen storage diseases. I think I covered this in part 2. Okay. So glycogen storage diseases, you just need to know the name. Okay, I think I have got a video dedicated to glycogen storage diseases where how you can remember the name of the enzymes. Okay, So yes, so we will basically stop here. If you want me to continue with glycogen storage disorders in a new video I can make, or you can simply search in my channel, you will get there's a very good video explaining how you can remember the glycogen storage disorders. What you need to remember, you need to remember the name of the diseases, the enzyme defects. Okay, mainly this is important, the name of the diseases and the enzymes that are defective. Okay, we are done with glycogen storage disorder, right? So in case of HMP shunt, what you need to remember is very selective. You need to study HMP shunt in a very selective way. You don't need to remember everything. This first step is an absolute must to remember. Glucose 6-phosphate is converted to 6-phosphogluconolactone by the enzyme glucose 6-phosphate dehydrogenase, that is G6PD. Why? Because this produces NADPH. I have already covered it in detail in my part 2 video. Okay, why you need to know about G6PD and all, you can have a look, but over here I'm just briefly recapping. This NADPH helps in formation of reduced glutathione, which prevents RBC membrane hemolysis. You can find the details in part 2. If you have seen that video already, then you are done. I think you can stop watching, right? And also you need to remember 2 more steps, that is the trans-ketolase reaction. Okay, this one, the trans-ketolase reaction. Okay, the trans-ketolase reaction, why? Because TPP is the cofactor, so vitamin B1 is involved, and from xylulose and ribulose it forms pseudoheptulose and glyceraldehyde 3-phosphate. So 2 5 carbon compound is forming 1 7 plus 3, so 5 plus 5. So the calculation of carbon atoms remains same. 5 plus 5 becomes 7 plus 3. If you look at the structure, you can see 5 carbon atoms here, 5 carbon atoms over here, 7 over here, and 3 over here. So total this is 10 in the reactant and product side. And also trans-aldolase reaction. In trans-aldolase reaction, this 7 plus 3 combines to form 4 plus 6, that is erythrose and fructose 6-phosphate. In case of applied question of HMP shunt, you need to know what are the important products from HMP shunt, that is purines, I mean the ribose, that is the 5 carbon compound that is utilized in production of purines and pyrimidines, that is DNA and RNA, and NADPH. So why NADPH is important and it's all, this is already discussed in part 2, and I think from here you can continue watching part 2. I'll also link the part 2 video in the description if you are seeing it for the first time. So I welcome you to my channel, and that's it for today. I'll see you soon with another video very soon. Till then, bye, I'll take care, and please let me know in the comment section if you liked this video, and if in fact you liked this video, and if you're watching this video till now, type the code of the day, that is HMP, in the comment section. If you type HMP, I'll know that you have watched this video, and that's a token of respect for me, and I love you for that. I'll see you soon. Bye and take care.